Alright, hello and welcome everyone. In this particular video, I am going to discuss about the process of initiation in DNA replication. DNA replication in eukaryotes. And here I am taking the example of yeast cell. Alright. So as compared to the prokaryotes, eukaryotes are higher living organism, right? So all the machinery like replication is also very much sophisticated in eukaryote as compared to prokaryote. So in all the organism, like in case of mammals, the process of initiation, how it begins is not much known. It is known, but it is still controversial. But the best example we are learning about is yeast, because yeast is also a eukaryotic cell, right? So if we study in the lower living organism, in the lower living eukaryote, the process of initiation of DNA replication, then it might be somewhat similar to the higher living organism also. They might also follow the same rule, and it has been stated that there is some similarities between the replication of initiation in the yeast and the higher eukaryote. So we, while studying about the replication in the eukaryote, we mostly take the example of the yeast cell. So let us come to the point. Now this is the ARS, or ARS is known as the autonomously replicating sequence. This autonomously replicating sequence is same as ORIC in E. coli. Alright, it is same as ORIC in the E. coli. This whole sequence is autonomously replicating sequence, and within this autonomously replicating sequence there is domain A, B1, B2 and B3. Alright, within the autonomously replicating sequence there is domain A, B1, B2 and B3. Now let us see what happens here. This domain B1 and domain A is known as ORS. ORS means origin recognition sequence, because these two domains, these sequence are responsible for the recognition of the main initiation point. Alright. Likewise, in E. coli we have origin recognition sequence. In the same way, here also we have origin recognition sequence. Now first what happens is the ORC. ORC refers to origin replication complex. Alright, origin replication complex. It is a protein. Origin replication complex protein will bind to ORS, that is in this particular region. First ORC will bind to ORS. As the ORC will bind to ORS, then what happens in the another domain, in the B3 also autonomously replicating factor binding protein. Alright, ARS binding factor we can tell. ABS means ARS binding factor. In the domain B3, ABF, that is ARS binding factor 1 will bind. As the two protein binds at the two end, after that what happens? The CDC6, CDC1 and MCM, they will bind to the ORC complex. Alright, first the CDC6 will come and bind, and then CDT1 and then MCM. Alright, this is CDC6, this is CDT1, this is MCM. After the binding of ORC, in the next step what happens is, now like this sequence will be recognized by the other protein, right? So now CDC6, CDT1 and MCM will bind. After they bind, what happens is that there will be the melting, because this part is AT rich, right? In the E. coli, here also the AT rich region will start to melt, okay, because of the torsional hindrance or some kind of stress, because in both the side the protein has been loaded, so there will be stress in the AT part, and since it is easy to break the double bond, the AT region over here will start to melt. And in the next diagram, what happens is the AT region has melted and they has formed a replication bubble. As the replication bubble is formed, the helicase will go and sit in the each of the fork. Alright, helicase will go and sit in the fork where the process, where the unwinding of DNA will take place. From the fork, unwinding of the DNA will take place. So helicase will come and sit over the fork, and then CDC6 and CDC1 will fall off. Alright, and the other protein is another enzyme, that is main enzyme, that is DNA polymerase, will come and sit for the process of replication to begin, and they form a repliosome. Right, repliosome complex is formed now. Primase, helicase and polymerase will go for the process of elongation, right? Other protein will fall off, and these helicase is nothing but MCM protein. Alright, here the helicase is MCM. Now one more important thing is CDC6 and CDT1 is an important protein. Here we are just watching it as it is coming, and then it is also recruiting the helicase, and then it is going off, right? But it has a major role to play. This process happens in the S phase, right, in the synthesis phase. But these two protein, what happens is these two protein will disintegrate as soon as the synthesis phase or the S phase will stop. This is because this protein makes sure that the replication does not initiate after the S phase. Alright, because say we in the cell cycle we have G1 phase, S phase, G2 phase, right? And then the mitosis or meiosis will take place. But during S phase, what happens? During S phase, what happens is there will be the synthesis of DNA. Synthesis of DNA means replication of DNA, right? So these we are studying about the initiation process of replication. So these initiation should also be happen during the synthesis phase of DNA, right? It will initiate and then it will synthesize. So these protein, these CDC, CDT6 and CDT1, this protein will be remaining in the cell only till S phase. As soon as the S phase will finished, this protein will be disintegrated by the enzyme. Alright, it will be disintegrated, and then therefore this marks that there will be no confusion during the process of cell cycle. As the cell will proceed, there will be no further initiation, because these two protein are the major protein which is helping in the process to bring out the process of initiation, and in the absence of this protein there will be no initiation. That means now there is no mistake, right? If the initiation will begin after S phase, then it is likely that there will be mistake. Some kind of cell may turn into the cancerous cell. But since there is the protein CDC6 and CDT1, which is the major initiate protein which is required in the process of initiation. That is why after the disintegration of this protein, there will be no further initiation, right? So let us recap what we studied today. In the yeast cell, in place of ORC, in the E. coli we have ARS, that is autonomously replicating sequence. And then we have the 4 domain A1, B1, B2 and B3. A and B1, they make a origin recognition sequence, and then here the first protein complex, that is origin replication complex protein, will come and bind. After binding of origin recognition complex, ARS binding factor will bind to B3. After binding of ARS binding factor to B3, now CTC6, CTC1 and MCM protein will bind to the ORC complex. As they bind to the ORC complex, the AT rich region will start to melt because of the tension created in the DNA strand. And since the AT rich region is easy to break, since there are only 2 hydrogen bonds, the AT rich region will break, and as the AT rich region will break, the MCM protein will shift towards the replication fork formed in the both the side of the replication bubble, and it will recruit. This is polymerase. This is polymerase, and it will recruit the polymerase enzyme as well as that is primase enzyme, forming a repliosome, and then the process of replication will begin. Another thing, this protein is known as origin replication complex, is because this protein is found to be binded in the DNA throughout the process, throughout the regulation process or the synthesis of the DNA. That is why it is known as origin replication complex. It is also helping in the origin also and also in the replication. Alright, this was only for today. If you like my video, just like, subscribe and share the video, and then I'll be bringing the next video on the same topic for the elongation. Thank you.